It's really a pleasure to be here and to have this opportunity to share with you a little bit the story of our PGM in the lab. So yeah, as you see from the title, what I will be talking to you today is how we approached moving our gene panels from Sanger sequencing to the Ion Torrent PGM. And the story, it's not that big actually. It didn't start long ago. So we began thinking about this issue in the beginning of the year 2011, and it was by the summer of last year that we got the PGM working in our lab. We spent the next few months trying to get a feeling for the technology and to see how things worked in our hands after having the training in Darmstadt. And since then, we've been slightly and consistently moving different panels that we had running in the lab to the Ion Torrent PGM. And those you can see on the slides. So actually what I will talk today is to tell you a little bit our approach and how we did this shift from Sanger to the PGM Ion Torrent. So the panels that we started with were essentially 3 panels. One that we used as our first testing panel was the BRCA1 and BRCA2 genes. Those are 2 genes that are very particular and have a lot of challenging characteristics that we wanted to see how the technology could address the sequencing of the 2 genes. And then we added more and more complexity to our panels. So we second moved to a gene panel that was composed of 8 genes, which was a big step, starting from 2 to 8. And lastly, we moved to the resopathies panel, where we have a total of 11 genes that we screen for different mutations. So our first question was, having this shift in the paradigm of sequencing, that how would we get our amplicons? What kind of strategy would we have to employ to enrich for the genes that we were trying to study? And by then there were clearly two ways of doing this. Was either using PCR-based methods or hybridization-based methods. We looked at pros and cons of the different technologies, and nowadays you even have a lot of commercial technologies that you can use to do this type of enrichment. But actually we went for the most basic one. So we approached this the most simple way that we could. So we used just regular PCR and adapted a little bit the multiplex PCRs that we had for our panels and adjusted it slightly to get good reads on the Ion Torrent PGM. So this resulted in the 3 panels, the completely different amount of PCR reactions that we had to do. You can see in the bottom of the slides, you can see the amount of amplicons that was involved in each of the panels, ranging from 77 on the BRCA1 and 2 to the resopathies with 123, and this was reflected in the number of multiplex reactions that we had to perform, from 14 to 17 reactions, all done in parallel and tested in parallel. So, very simply, this was actually the major shift that we had to do on the procedure that we used to follow, was that after isolating genomic DNA and performing the different multiplex reactions, instead of splitting all the different amplicons and performing the sequencing on the individual amplicons, we did opposite. We mixed all the multiplex reactions in a single tube and used that tube for entering the standard protocol for preparing samples for the Ion Torrent. So we do enzymatic fragmentation of our samples. I didn't have a picture here, but the fragments on the amplicon size that we had on our reactions ranged from 215 to up to 600. So in fact, we had to fragment our DNA. And then we followed the phosphorylation, adapter and barcode ligation to the sample so that we can run multiple samples per ship. Then prepare a template with the one touch, which was really a big improvement in the initial manual protocol of doing the emulsion PCR, and then doing the sequencing on the PGM. Then follows the data analysis. This was something that we really had to come up from the beginning. This is totally different from the pipeline that we had for Sanger sequencing. So we use most of the functionalities that the torrent server has for selecting the good quality reads and trimming the adapters and splitting the barcode files into different files. We upload the FASTQ files into the segment engine from DNA Star for assembly and alignment. And then we use the Segment Pro for variant filtering and coverage and variant analysis studies. Then we do more manually and more visually work to identify known SNPs and known polymorphisms and to validate the variants that we detect. And this validation, we still use Sanger sequencing to validate all variants. So in order to have this shift running routinely in the lab, we had to make a validation study to see how powerful this technology was. And for this, we selected 110 samples from the lab and divide them into different groups. One group, which I called it here the training set, was composed of 35 samples for which we had previously fully sequenced with Sanger sequencing, and we knew beforehand the known variants that were present there. And this was including a lot of different positive cases that we had, involving many different types of mutations, from missense, deletions, large insertions, and gains. And then we used a second set of samples, composed of 75 samples, for which we had a priori no knowledge of the sequence whatsoever, and we ran them in parallel, using both the Ion torrent procedure and the Sanger sequence. And in the end, we did the comparison of the outputs of the two studies. In the bottom of the slide here, you see that for this study, we basically use only the 314 and the 316 chips. We haven't run yet a single 318 in our lab, and I think it won't be that, it will take a lot of time to run one of those. We need a lot of throughput to get a lot of samples to be run at the same time on a 318. But at least you get an idea here of how many samples one can multiplex in a single chip and run them at the same time. So from the training set, we use this training, since we knew what variants were present in this training set, we use this set to adjust mostly our data analysis, how to set all the filterings in a way that we knew that we weren't losing any information that we knew was relevant. And at the same time, we also started to get a feeling of what we would consider a good run or a bad run, depending on the number of reads and the Q20 bases that we would get out of 314 or a 316 chip. Another aspect that was really important here at this stage was that we were still optimizing our multiplex reactions for the Ion torrent. So it was important for us to, in all the runs, to check if we had coverage of all the amplicons that we had in our multiplexes. And if they were not present, then we would keep note of those and later on use Sanger sequence to run those amplicons that were not covered. So after getting all our tools well defined and well oiled, then we moved into the validation set, where, as I told you before, we did the multiplex PCR reactions for all these samples, and then we did the analysis in parallel, both using the Ion torrent and the Sanger sequencing. And here, this cartoon here is just for those of you that don't do this routinely in the lab, to get an idea how much different is the amount of work that one needs to do using one or the other technology. So if you compare on the left side, you have the procedure for the ion torrent. You see that in a single chip, you can, in this case, run up to 5 patient samples at the same time for all the amplicons that you are, for all the genes that you are studying. And on the right side, if you're doing Sanger sequencing, you need to do, in this case, a lot of single reactions just to study one of the patient's material. So this was really one of probably the biggest advantages that we saw from moving to this technology, was to get less work on the workload on the technical staff of the lab. So as I told you in the beginning, it was really important for us to be sure that we are not having any false negative results. So, and this could easily be having false negative results by just not having amplicon coverage. One of the amplicons during the PCR reaction was not being amplified, and we would be losing information there, and we would call this a negative, and yeah, it was just because we didn't have any information there. So the first look that we have on our samples, on our runs, is to get a feeling of the coverage of all the different amplicons. And this picture here just shows you that what we are really sequencing is, it's a pity that I don't have a pointer so that I can show you. But on the red bars are the distribution of the depth of read. So the higher, the more reads you have on that particular region. And on the small green bars on the bottom represents the exons of this particular gene. This is, I think, the end of BRCA2. And you see that what we are sequencing is essentially only the exon part of the genes. And if we had the missed amplicon here, we would clearly see by a lack of representation of reads on that region. And this is illustrated here. If you look on BRCA2, this is the exome of this gene that we are sequencing, and the orange arrow points to a particular amplicon that for unknown reason was not amplified during the PCR reaction. And what we had to do later on was to, in the end of this analysis, was to pick up those primers for that region and sequence them on Sanger sequencing. So from this picture here, you also see that we were not very much worried on how really uniform this amplicon coverage was. We were more worried to not have amplicons not being covered instead of having them all with uniformity coverage. So now I'll just go quickly on 3 slides where I show some positive results, some of the data that we got out of this. And here is just for you to have a comparison of the type of information that one gets from the readings on the ion torrent PGM and the Sanger sequencing. And this is just to show homozygous alteration on the BRCA2 that is clearly pointed out by the data analysis and confirmed by Sanger sequencing. Here you have an heterozygous alteration that was also clearly seen in Sanger sequencing, and we had a very balanced alteration also on the ion torrent PGM. And for us, what was what we had less expectations was to find small deletions and small insertions. And in this case, I think at least the people in the audience here won't see it very clearly. But where the arrow is, there's a 4 nucleotide deletion that it's present in almost 50% of the reads. So this gave us confidence that also small alterations could be detected using the ion torrent and the data analysis pipeline that we were running. So in the end of the old analysis, we compiled the entire data that we got, and this resulted without using any filtering from our data analysis, resulted in 4,912 variants that were determined by the ion torrent PGM. And after performing all the data analysis, according to our pipeline, this resulted in a false negative rate of 0%, which is extremely important for us. We do not accept any false negative in our data as much as we can. The true positives, we identified old variations that were identified by Sanger sequencing. We also identified them by the Ion Torrent PGM. We eliminated 4,402 negatives. These were mostly calls that had poor quality, so they were easily eliminated. And we detected with the Ion Torrent, or the Ion Torrent PGM resulted in 59 false positives that were eliminated using the Sanger sequencing. So in the end, doing overall calculation for this approach, this resulted in a methodological approach that resulted in analytical sensitivity within a 99% confidence interval of 99%, and analytical specificity of 99%. So we were pretty happy with this result. So just a quick summary of how we approached all this shift from changing our routine work from Sanger sequencing to the Ion Torrent PGM. So basically, we followed the entire procedure using the sample preparation for the Ion Torrent. All calls that are not filtered away during the data analysis pipeline are validated using Sanger sequencing. Amplicons that are missed during the amplification are also validated using Sanger sequencing. And just to give you a number of how much this results in the end, this represents around 1.6 Sanger sequences that one needs to do per sample, which is relatively very low. So I think this clearly points a way that for us it was simple, practical, and I think it's a valid approach for this type of studies. So this was just not my work. This was mostly done by different people in the lab. They are here pictures. Sonia Souza was involved in the BRC2 panel, Sheila Ribeiro for the HCM, and Ana Justino, which is here in the audience if you want to discuss, was involved in the setting up the rosopathies panel. As Aldo pointed out, I'm the local here, so you are all welcome to visit us 600 kilometers up north from Porto. The weather is not as nice. It was raining this morning, so I'm not sure if you will make that visit, but you still, anytime you like, you are welcome to visit us. So thank you very much.